This video will discuss mutations with a quick overview on the central dogma and interpretation of the genetic code. There will also be practice questions at the end of the discussion. There are timestamps in the description box below if you want to skip to a particular topic. Okay, so quick review muna tayo. DNA to DNA is replication, DNA to RNA is transcription, and RNA-directed protein synthesis is translation. But when do mutations happen in the central dogma? So this happens during replication. So pag may mutation sa DNA, matranscribe ito sa mRNA and thus cause changes in the encoded protein. So in short, there will be a domino effect. So quick review muna tayo sa translation. So bakit ba translation yung tawag? So protein synthesis is called translation because the language of nucleotide sequences na na-transcribe sa mRNA, which is yung adenine, guanine, uracil, and cytosine, these bases will be translated to amino acids, which is the language of proteins. So this leads us to the genetic code. So the genetic code is para siyang dictionary na nagtra-translate sa nucleotide bases to amino acids. So for example, uracil, uracil, uracil corresponds to the amino acid phenylalanine. There are 64 base triplets. Base triplets are called codons. So to summarize, 3 nucleotide bases is 1 codon, and 61 of these codons correspond to an amino acid. So how do we use the genetic code? So for this example, GUU, we go first to the left side for the first base, G, and then to the upper side for the second base, so U, and if you refer to the left side, U, GUU. So GUU corresponds to valine. So you do not need to memorize all of this. Usually sa board exam, magbibigay sila ng genetic code just in case they ask you to translate. However, you should take note of the special codons. So these are the 3 stop codons and the 1 start codon. So for the initiation codon, this is AUG, and this codes for methionine. So how I remember this is the sentence, classes start in August. Now, as for the stop codons naman, my mnemonic for that one is stop, you are annoying, you go away, you are gone. So UAA, UGA, and UAG. These are all the stop codons. So now that we've reviewed translation and codons, we can now discuss mutations. So to reiterate, pag may mali sa DNA, may mali yung nasa mRNA, and pwede ding mali din yung lalabas na amino acid, which will cause problems sa resulting protein. So there are 2 broad mutation types. This would be the small scale or point mutations, the large scale, or frameshift. Okay, so point mutations occur when one base DNA is replaced by another. So pwede yung adenine maging guanine, thymine, or cytosine. So when a pyrimidine is changed to another pyrimidine or a purine is changed to another purine, this is called transition. However, if a pyrimidine is changed to purine or vice versa, this is called transversion. So obviously, magbabago na yung DNA sequence, so iba na din yung codon sa mRNA, which will lead to mutations during the protein synthesis. So these mutations are named based on their effect on the resulting protein. So there's silent, missense, and nonsense. So okay, first let's start with silent. So here in the example, you can see that guanine becomes adenine. There is a mutation happening. So this will affect the mRNA strand. Instead of cytosine for guanine, naging uracil na for adenine. So as I've said, may domino effect. So instead of cytosine in the original sequence, naging uracil. However, we can see that it is still the same amino acid, histidine. So if we look at the genetic code, CAC and CAU both code for histidine. So the amino acid sequence stays the same. That's why it's called silent mutation, kasi parang walang nangyari. So next is missense mutation. So missense mutation results in one amino acid being replaced by another. So if you look at the normal sequence, instead of guanine, it's supposed to be thymine. Now, because of that mutation, it will be passed down to the mRNA sequence, and this will also affect the encoded protein. So instead of histidine, dapat yung third position naging proline. So this is missense mutation. A very common example of a missense mutation would be the sickle cell anemia. So as we've discussed in an earlier video, instead of glutamic acid, yung nasa 6th position, it became valine. So nag-iiba na yung structure sa hemoglobin, which ultimately affects the red blood cells shape. Next is nonsense mutation. So if you recall, there are 3 stop codons. These are UGA, UAA, and UAG. Now, nonsense mutations happen whenever a mutation results in any of these 3 codons. So, if you look here, instead of adenine, naging cytosine, so the pair would now become guanine. So, this gets passed down to the mRNA, which ultimately affects the protein. So, instead of UAU, naging UAG. Now, remember that UAG is a stop codon. So, this ultimately leads to premature termination. So, mag-stop na yung elongation sa peptide chain, so naging shorten na siya at non-functional. Okay, so now that we're done with silent, missense, and nonsense mutations, we will now go to large-scale or frameshift mutations. So there are 2 types. There's insertion and deletion. Okay, so remember na yung codons ay base triplets. So they are read by 3. Now, if for example, during DNA synthesis, may isa or dalawang nucleic acids na mai-insert or delete, ang resulta nito ay frameshift mutation. So, makikita mo sa figure na may na-insert na base, this one in position 4. So, syempre this gets passed down during RNA synthesis and will cause problems during protein synthesis. So, makikita nyo na all the amino acids beyond this point ay mali na. All because yung reading frame is shifted to the right. Because may na-i-insert na nucleic acid. So, same din sa deletion. Only difference is may nawawala na base. So, yung mRNA code is different din. And during protein synthesis, maling-mali na din yung amino acids. So, all amino acids beyond this point are wrong na dahil lang may na-delete na isang nucleic acid. So, the reading frame is now shifted to the left, and because of that, mali na yung encoded protein. Okay, so practice questions. This type of mutation occurs when the codon containing the changed base may become a termination codon. Okay, so the answer to this one is nonsense mutation. Okay, so nonsense mutations result in a premature termination of the growing polypeptide chain. So remember the 3 stop codons UAA, UAG, UGA. The result of a point mutation that occurs when UCA, which codes for serine, is converted to UCU, which also codes for serine, is called Okay, so the answer to this one is C, silent mutation. So as discussed earlier, silent mutations do not affect the amino acid sequence of a protein. So kaya nga silent kasi parang walang nangyari because there are no changes in the amino acid sequence. 2-aminopyrine is no longer used clinically. It has been found to replace adenine for guanine in DNA, resulting to bone marrow toxicity. 2-aminopyrine induces what type of mutation? Okay, so the answer to this one is A, transitional. Okay, so as discussed earlier, if a purine is converted into a purine, or a pyrimidine is mutated into another pyrimidine, this is called transitional. However, if a mutation of a purine results in a pyrimidine, or vice versa, this is transversional. Okay, so thank you for watching, and I'll see you in the next video.